It's important for us to understand in science where we came from and sort of the development of our field. So as a background, I'm going to give you some information about some important people that helped to bring us to the point where we are in microbiology today. So I'm going to start by talking about people that are sort of pre the 20th century, so before the 1900s. So one of the first the birth of microbiology had to be the discovery of microorganisms. So developing the technology of a microscope in order to see an organism that tiny. So some of the earliest microbes were seen by Robert Hooke. He developed one of the first microscopes and used it to see the first cell. And this was in the 1600s. Now, similarly, Antonie van Leeuwenhoek, a little bit later in the late 1600s, he was a Dutch cloth inspector. So he actually used microscopes and lenses in order to do thread counts and determine value of cloth. So when he was doing this, he used this single lens microscope, and he was able to see these tiny little organisms. So he drew them. Okay, and this is actually some of his original drawings, much of like what we see under a microscope today. And he reported those findings to the Royal Academy of Science. So because he, had some of the first drawings ever of bacteria and protozoans, he is actually known as the father of bacteriology and protozoology. Now, some women were also involved in some early discoveries. So a British diplomat named Lady Montague was living in Turkey at the time, and worldwide smallpox was rampant. And so in England, men had these huge pock marks and sores, from having smallpox. But she noticed that men in Turkey did not have smallpox. So she looked a little bit into, why this might be so. And she found out that actually the Turkish people would take their children to witch doctors, and the witch doctors would take pus from smallpox and rub it on the children. And essentially what this would do was that it would inoculate them. They would get smallpox as children, which is fairly mild, it's non-life-threatening, and it wouldn't leave these giant pock marks that it did in other people or older people that got the disease and survived. So she, discovered what is called virulation, or inoculating someone in order to, give them an infection. So, it's interesting to think about that. A lot of religious beliefs, tribal, medicinal sort of things, actually are steeped in what we now understand as, some true science. So, we're jumping ahead now to the late 1700s. Edward Jenner did something pretty similar to Lady Montague in recognizing that milkmaids did not catch smallpox. So seeing that a certain group of people were sort of seemingly immune to a certain disease. And so he proposed that possibly the reason why the milkmaids didn't get smallpox is somehow they had cowpox, or they were exposed to cowpox, and the cowpox then prevented the smallpox. So he created a vaccine for smallpox using cowpox. So cowpox is similar enough that it produces an immune response in us and creates antibodies to smallpox, but it's cowpox, so it does not make us sick. Okay. Now, again, remember, these people are just experimenting. They're not really doing things the way that we understand them to work now. They were just trying, different techniques, and they didn't really understand why they were working. Okay. Now, in the 1800s, Ignaz Semmelweis was one of the first people to sort of start to develop this idea of thinking about the spread of infection. And he was a physician, and he noticed that women who were giving birth with midwives were not getting childbed fever, whereas those that were having children under the care of doctors were getting childbed fever. And so he noticed the difference is that midwives would wash their hands and the doctors did not, because it was sort of beneath them to think that they were dirty in some way. So he proposed that by washing your hands you could prevent childbed fever, and for doing this he was actually shunned by his doctor friends for proposing that they somehow were making their patients sick. Right around the same time, Louis Pasteur, who was a French chemist, started making some of the most important discoveries. He's one of the most important influential characters in this time period of microbiology. And he had this famous quote that's, chance favors the prepared mind. So by recognizing, kind of going on those ideas of those previous people, by recognizing sort of correlation between one thing and another, or errors that happen, and seeing how they positively affect our situation. And I'll give you some other examples of that later too as well. So one thing that he came up with an idea is he opposed the idea of spontaneous generation. Well, spontaneous generation was the idea that people had commonly at that time, that bacteria formed from non-living materials. And the reason why is because, you would set out a piece of bread and mold would start to grow on it, or you'd leave water out and it would get cloudy and bacteria would grow in it. And so how did the bacteria get there? And because we couldn't see them going from one place to another, people thought that they produced out of sort of thin air, out of non-living materials. But so he did some experiments to show that organisms actually traveled through the air, and he did this by filtering air and viewing those organisms. Okay, so then it was an idea of trying to work on how to kill them, and how to prevent them from contaminating things from moving from one place to another. So he came up with the idea of using heat to eliminate living things, and he was able to do this except for a special type of organism or bacteria that forms a spore. A spore is a highly resistant sort of egg of sorts that can hatch into a bacteria that's formed by some bacteria in under stressful conditions. He also created something called the Pasteur flask. It's a swan neck flask like this, and just the shape and the angle of the flask helped to prevent the bacteria from contaminating what was ever in your flask. Some other major discoveries, and I'll talk about some of them later too, is he developed vaccines for anthrax, cholera, and rabies, and he was the first person to suggest germ theory, that microorganisms were actually spreading disease. So Ignaz Semmelweis had the idea of washing your hands, but he didn't really understand what that meant. So Louis Pasteur proposed that killing microorganisms prevented the spread of disease. Now, John Tyndall worked with Pasteur, and he was able to develop a method called Tyndallization, and this was a way of killing those spores, so those heat-sensitive spores that form under poor conditions. And he did this by a process of boiling and incubating and boiling and incubating. So you would boil and spores would form, and then you'd incubate and you would kill them, and then they would hatch, and then you'd boil again and it would kill all the organisms that you had in there, and then you would incubate again just to assure that everything in there was dead and that whatever you're working with was sterile. So since Tyndall and Pasteur came up with this idea that germs spread disease, Joseph Lister decided that he should develop methods to try and prevent this from happening in medical practices, specifically in surgery. So he came up with the idea of antiseptic surgery. So as we're doing surgical procedures, taking measures to prevent the spread of disease by using gases around the wound, by washing hands. So these are all examples of things that they did to start to prevent the spread of disease by microorganisms. So with the idea that microorganisms cause disease, Robert Koch came along, and he decided, well, let's try and prove this theory, and if it's true, let's link certain organisms to certain diseases. So he started by studying anthrax, and what he determined was that any animal or person that was infected with anthrax had the bacteria Bacillus anthracis. However, he had to come up with a method to show that the Bacteriobacillus anthracis was actually the cause of the disease and not a result or like a side effect or something that was happening as a result of the disease. So he did this by injecting bacteria into healthy organisms. And by doing this, he developed what is very important and we still use today in order to link Koch's postulates with, to link a certain organism, microorganism, with a disease. So this is to prove what organisms cause what disease, and it's a 4-step method. So the first thing that you need to do is ensure that whatever bacteria or microbe that you're trying to link to the disease is constantly present in animals that are sick and is not present in healthy ones. Then you isolate that organism from a sick individual and you grow it in a pure culture in a lab. Then you introduce that microorganism into a healthy, susceptible individual that then the disease is caused. And then the fourth thing is then to re-isolate it and show that you still have the same organism. So pretty much everything follows Koch's postulates, but there's a few rare diseases that don't, like AIDS, gonorrhea, and this typically has to do with the inability to culture them very well outside of the organism, or for them to not be able to live outside of the organism. So some of the major diseases at the time that Koch worked on were tuberculosis and cholera, which killed huge numbers of people. And he discovered the bacterial strain for tuberculosis, which is what won him the Nobel Prize. And he also found what caused cholera and linked it to water sources, so was able to prevent the spread of cholera by, telling people to filter water or use, clean or different water sources. Okay. Now, speaking of cholera, I want to go back and actually talk a little bit about Pasteur, because this was another sort of major discovery at the time. So, again, this is a chance favors the prepared mind idea. Pasteur tried Koch's postulates with chicken cholera. So he's trying to prove that chicken cholera was caused by a specific infection. So part of it is to grow a pure culture out of the lab. So he took an old culture and he left it, or he took a culture and he left it in the lab for a significant period of time. And when he went to re-inoculate his chickens, his healthy chickens, to make them sick, he used a very old culture that had been sitting on the shelf for a while, and he noticed that the chickens didn't get sick. So he tried it then again with some fresh cholera, and again the chickens didn't get sick. So by doing this, he actually inoculated the chickens with what is called an attenuated or non-virulent, non-disease-causing microbe, and this is how he discovered vaccines. Alright, so we will continue in the next lecture and talk a little bit more about vaccines that have discovered in some modern technologies in microbiology.